students, so this video is about cell division and the cell cycle. So by this end of the video, we should understand the major events that happen in the cell cycle and then eventually how the cell divides during the process of mitosis. So I'd like to begin by telling you a story. Once upon a time, there was an egg and a sperm, and eventually they came together and a zygote was formed. That zygote had all the genetic information that cell would need, and then that cell divided. Those next two cells have the exact same DNA. Then they divide again. Eventually, you get an embryo. Every cell in this embryo has the exact same DNA. However, I see there's an eyeball forming, there's a brain forming. Differences exist because different genes are expressed in different cells. However, in order to get from a single cell to an embryo or to get to a full human being, or whatever organism it is, cell division has to occur. So we use mitosis for a bunch of things, like this, embryonic development, tissue repair if you ever injure yourself, asexual reproduction, growth. All of these things require the cellular division known as mitosis. So let's break it down. There's going to be a bunch of different things that happen during mitosis. In order to really take a cell and divide it into two, then all the organelles need to copy, etc., etc. But let's focus in on just the chromosomes. So the first thing that's going to happen is the DNA, which is sort of fluffed about, is going to be condensed into chromosomes. That's going to allow them to divide without getting tangled and to make sure that every daughter cell has the exact same amount of DNA. Interphase is the part of the cell cycle where a cell is being a cell. Interphase is broken into 3 steps, G1, S, and G2. During S, the DNA is copied. So let's go through the steps of mitosis quickly. Here's prophase. The DNA has already been copied. And then the cell is going to break down, the nucleus I mean. Here are the chromosomes in the middle during metaphase, separate during anaphase, form new nuclei during telophase, and then the whole cell splits into cytokinesis. So let's break down each one of those steps a little bit more. First, here's an image of a cell. This cell is currently in interphase. As I look at this cell, there's some things that I'm going to see. The cell is going to be doing what a cell does. It's going to be growing in size, it's going to be doing protein synthesis, and eventually it's going to get to the S phase where it copies its genetic material. This is what a cell might look like. We see our cytoplasm, our membrane, we see this nucleus, but there's nothing really defined in the center. It just sort of looks like squiggles so far. That is all of the DNA just fluffed about. There's actually a lot of stuff happening during interphase, and I cannot underestimate the importance of interphase. During G1, that is the biggest growth phase. Right before the DNA is duplicated, the cell is going to be growing and being a cell. During S of interphase, the DNA is duplicated through DNA replication. During G2, the cell prepares for division by making sure there's enough organelles, etc. Then we enter this phase of mitosis. This is an image of a cell during prophase. Notice that the nucleus, where it used to all just be sort of undefined, is now forming these little strands. Those strands are the chromosomes. We call those like chromosomes condensing. The nucleus starts to swell. The nucleolus, which is in charge of making ribosomes, starts to disappear. Eventually, the envelope for the nucleus is going to break down, and spindle fibers are going to form. The chromosomes are going to appear as 2 threads. Sometimes if you get a really good image, it kind of looks like this X. They're attached in the center by a centromere, and each side is called a chromatid. Each side is a copy of the DNA. So here's 1 copy and 2 copies. They just stay together at this point to make sure that they're evenly broken down. Here's an image of a cell that is in metaphase. Notice those chromosomes are in the center of the cell. The nucleus now is gone. The spindle fibers are pulling on those chromosomes until they line up in what we call the metaphase plate, the center of the cell. Eventually, those chromosomes are going to start to separate. That process, as they separate, we call that anaphase. So we have the sister chromatids of each chromosome being split apart. So we have half the DNA going this way and half the DNA going that way, and the spindle fibers are going to be what pulls that DNA to each pole. I think of spindle fibers kind of like fishing line. So here's the fishing pole, it attaches, and then it pulls the DNA back towards the side. Here's an image of a cell when it starts to divide further. So So this is after anaphase, and two new nuclei are going to start to form, and the cell itself is going to start to divide. The process of the nuclei forming is called telophase, and the process of the cell dividing is called cytokinesis, and generally they happen at about the same time. During this stage, the nuclei are going to reform. Each nuclei is going to have a complete set of chromosomes. A new nuclear envelope is going to form. Chromosomes are going to uncoil, and eventually we're going to have two daughter cells that are exactly like the original, and the cell is going to go back into the cell cycle, again in interphase, to do its job as a cell. So let's review that just really quickly. If we're looking at the chromosomes and where they are doing, or what they're doing and where they are throughout each type, we have multicellular organisms. They're going to copy their chromosomes before cell division so they can grow to a mature size. All of that's going to occur during interphase, where the cell is being a cell. Then during mitosis, the nucleus itself is going to divide in the process of prophase, metaphase, anaphase, and telophase, distributing those chromosomes into two equal groups. Then eventually the rest of the cell is going to divide during cytokinesis. Sometimes cells are not going to actually divide anymore. If that is the case, they go into something called G0 or G0. That is going to be a cell that stays in interphase forever. Some of them can stay in that way for over a year. Some are never going to go back into interphase. So a lot of people want to know, okay, how long does that happen? How long does it take for that process to occur? And the answer is it really varies a lot depending on the cell type. Some cells are able to go through a lot of cycles in a short amount of time. Some cells take a really long period of time in order to go through these. So if we look at some of this data, bean root tips, we notice those have this many cell cycles per hour, meaning they can grow fairly quickly. They divide fairly quickly. Versus if we looked at something like a hamster fibroblast, we see it goes a lot slower. So depending on the cell, we're going to have a lot of differences in how quickly cells can regenerate, or how quickly cells can divide to regenerate tissues. There's some consequences of problems with the cell cycle. So if we've got the cell cycle going through G1, S, G2, and then mitosis, there's actually going to be little checkpoints at the end of each one of these stages. Those checkpoints are going to go back and they're going to check for any problems. So at the end of G1, there's a DNA damage checkpoint, where it goes back through the DNA, double checks the DNA is working well. After S, we have another one of the checkpoints that goes back through the copied DNA to check for any issues. At the end of G2, we have another one, another DNA damage checkpoint and a DNA replication checkpoint. And then at the middle of anaphase, we have another one. We have all of these different checkpoints throughout the cell cycle. The reason for that is because if there's an issue with the cell and the cell cycle gets messed up and the cell cycle starts to go too quickly, or the cell isn't spending enough time in G1, isn't spending too much time in M, then we end up with tumors. So normal cells are going to have controlled cell division, where division is going to eventually stop and it's only going to go through this next stage if it's past this checkpoint. However, if you have a cancerous cell or a tumor cell, it's just going to keep dividing in a rapid rate. Notice there's some differences in the shapes of these cells. The reason for that is because cancer cells are just dividing, and so they don't spend the time necessary in G1, S, and G2 to make sure that they're a fully functional formed cell. That happens with issues in the DNA. Each one of these checkpoints is controlled by genes. Some of those genes, we can call them tumor suppressor genes. And so normally if there's a tumor suppressor gene, if something happens in any of these checkpoints and a cell starts to divide in an uncontrolled way, then it will go through this process called apoptosis where it kills itself. However, if there's an issue, if there's a mutation in that gene, then this non-functioning protein is going to be made and the cell won't be able to regulate its cell cycle and it's going to form a tumor. Cancer is obviously a really big issue for a lot of people health-wise, and so understanding the implications of mitosis in that way, pretty important. Here's another way to look at that in just a little bit more detail. Here it's showing some of the specific things involved, like G1 cyclin, something called these kinases, CDK. If you get into cancer and want to learn a little bit more about it, you're going to see these things over and over again. Just know those are issues with the checkpoints. Because if there's issues with the checkpoints, that's how the cells start dividing in an uncontrolled way. If you want to go even a little bit more detailed, you can see here's a bunch of the specific proteins that are involved in each one of these checkpoints. We have growth factors that are balanced by tumor suppressors. We see all of these different genes. If you want to do cancer research, you'll probably be working on one of these genes. Okay, so now let's assume we do have a normal cell again, and that cell is growing, it's dividing, it's going through mitosis. Not every cell looks the same, but we started as one cell, and so let's think just a tiny bit about how cells differentiate. So cells in all multicellular organisms are going to differentiate to carry out specialized functions by expressing some of their genes and not others. Active genes are usually packaged in that expanded, accessible form of DNA, where it's not super tightly packed. That form is called euchromatin. Inactive genes are going to be packaged in a very condensed form. They're going to be wound around proteins. Those proteins are going to be wound around themselves again until the chromatin is very condensed. We call that heterochromatin. For example, if you have a single cell and you have two identical daughter cells, if a certain gene is exposed, then it can form an epithelial cell, like a skin cell. If a certain gene is exposed, a different gene is exposed, then you can form a neuron. The difference between these final cells is really just dependent on whether or not different genes are packaged in euchromatin or in heterochromatin. There's some differences between euchromatin and heterochromatin that have to do with methylation and acetylation. Just want to briefly point these out, even though it's a little more advanced than we need to go into. So if we look at the ends of these little yellow things, those yellow things represent histones. Histones are a type of protein that are going to associate with the DNA. The DNA are these little tiny threads sort of wrapped around those. So we see the DNA itself is wrapping around those histones, If we have these little acetylated histone ends, then the histones are not going to get too close together. That is going to allow this to be copied. But if we go over here and we add a methyl end to the end, notice how these change from this sort of squiggly version to a hook version? That represents these histones associating more closely together because of a methyl group that's been added. That is going to form the heterochromatin. So whether or not these things are added is going to depend on whether or not these genes can be expressed. Stem cells are an example of something that has not been methylated yet. It's unspecialized cell. They have two main key qualities: self-renewal and potency. So they can divide into different cell types, and they can continually divide and replicate. They can be derived from embryos, from the placenta, from the umbilical cord of the mother. We also can get some stem cells from adult tissue. Sometimes they're like slightly stem cell instead of a full stem cell. And there's a lot of possible implications of stem cells. So here we have some stem cells, and they can form muscles, blood, neurons. They have the ability to differentiate into different things. Some uses, possibly, I mean, there's so many ideas. Some of the things that I've seen so far are things like retinal cells, which are in the eyes. There's been a lot of research on stem cells to try to replace some of those. A lot of research on trying to do grafts for people who have been damaged in fires. A bunch of stuff with nerve cells, especially challenging to have any sort of regeneration of nerves. And so looking at spinal injuries, all of that's a possibility with stem cells. So in the future, I think we'll see a lot of research about stem cells. Okay, with that being said, that was the process of dividing our cell into two through mitosis and a little bit about differentiation of cells.